All right, so in this video, we're going to take a look at some of the topics from the nucleic acids section. And the first one here is recognizing nucleotides. And as a quick reminder, when we look at nucleotides, which are the building blocks of nucleic acids, those are made up of your sugar connected to a nitrogen-containing base, along with a phosphate group. So if you think of sort of a block diagram of what a nucleotide is. All right, we're going to have our sugar connected to our phosphate group, which will represent as P. And then we have our base over on the other side. All right, so all three of them make up a nucleotide. Nucleoside is just a sugar and the base itself without the phosphate. So as you look through the different options here, you need to think about, well, what components do I see? Well, in this first one, it looks like there's a sugar. Remember, the sugars are going to be in the ring structures because those are most stable. We have a nitrogen-containing base here on the right. And on the left side, we have our phosphate group, right? So that would be classified as a nucleotide. Same thing, one here down at the bottom. We have a nucleotide. As you go down, look at the last one, you'll see there's our sugar, there's a nitrogen-containing base. We don't have the phosphate, so that would be an example of a nucleoside. Another thing to keep in mind is if you're not sure whether the structure drawn on the right is one of the nitrogen-containing bases, so whether it's A, T, C, G, or U, you can always go to your data tab here on the right, scroll down to the, let's see down here, the nucleobases, and it will give you the structures of these, just to confirm that there are actually bases that you'd find in a nucleoside or a nucleotide. All right, so in this one, you're given a scenario of having alien life form that has DNA, just like human DNA, except that this other form of DNA is made up of deoxyarabinose compared to deoxyribose. All the other components are going to be the same. All right, so typically in DNA, right, we have our DNA nucleotide. We have our phosphate connected to our sugar. The sugar is deoxyribose. And then that's connected to our nitrogen-containing base on the right. So in this alien DNA now, the nucleotide in this case is going to have all the other components be the same. So we have our phosphate, we have our base, but now instead of deoxyribose, we're going to have deoxyribinose. So we need to figure out what that structure is. And in the data tab, you'll see that a bunch of these different structures are provided. For example, we have our simple sugars here, and you can scroll down and see arabinose and ribose are actually right next to each other. So I've copied and pasted that onto our page up here at the top. So you'll see both of them have the same structures. The only difference is on carbon number 2 in arabinose. The OH is on the left side. On carbon number 2 on ribose, it's on the right. Everything else is identical. So these are actually isomers of each other. So when they form the ring structure, they're going to be look the same with the exception of where the OH is on carbon number 2. So remember, when these ring structures form, it's the hydroxyl on the penultimate carbon, carbon number 4, that reacts with the carbonyl on carbon 1. So we have our 5-sided ring that forms. Let's see if we can get our structures here. So if you look on your toolkit here on the right side, you'll see some options for drawing your structures, and it has these templates already built in. So you can click on the sugars, and you can see you have your options for the Hayworth projections, the Fisher projections. You want to select the one that has a 5-sided ring, the Furiner's ring, and we can add that in. And if we look at our carbons now, we can sort of relate that to the carbons that we have in our sugars, right? So this is going to be our carbon 1, that's carbon 2, carbon 3, carbon 4, and then carbon 5 is going to be jutting off of the ring, right? And that's how ribose would look in the ring form. That's how arabinose would look on the ring form as well, because they are identical to each other. So then we can go ahead and we can draw the H's and the hydroxyl groups in. All right, so I have the straight chain formula shown here, the Fisher projection, and then we've just converted that over to the Hayworth structure, the 5-sided structure. And just to orient ourselves, the carbon number 1 is going to be this carbon here on the right. Carbon 2 is this, carbon 3, carbon 4, and then carbon 5 of the CH2OH is coming off of the ring. So now we need to put our OHs in. So let's start down here with carbon number 2, has an OH to the left. That means the OH is going to be pointing up. Carbon 3 has an OH to the right. That's going to be pointing down. And then the OH here on carbon 4, that was the one that reacted with carbon 1. So there isn't an OH here as well. That's where it was. Made a new bond to carbon 1. All right, so this would be arabinose in the ring structure. However, this is deoxyarabinose, right, which means that there's an oxygen missing from it. And just like in the case of deoxyribose, that oxygen is not going to be present on carbon 2. So instead of that being an OH, that would be an H over here. Okay, and we can complete the other H's, put those in up here as well. All right, so you have a basic sort of central part of this particular nucleotide. Let's go ahead and attach the phosphate group here to the right. So remember, phosphate is going to look like this. So we have your phosphorus connected to 4 oxygens overall with a -3 charge. So we're going to connect this one to this carbon here. Okay, and as a result, we're going to lose that the hydroxyl group, right? So we're going to connect it via a bond. It's going to be a phosphate ester bond. So there's my O to the P, and everything else is going to be the same. These 2 oxygens have negative charges, and then I still have my CH2 on carbon number 5. So that's my sugar. There's the phosphate. I still need the base. Well, which base do we need? Well, it looks like we need guanine, right? And we can go into our list, our data, and locate the structure for guanine. You don't have to draw that from scratch. That's actually already built in over here. All right, so you can copy that over to, or at least draw that over here on in your structure. So we're going to connect it via or to this carbon number 1, and that's connected to that nitrogen, and we can complete the structure for guanine here on the right side. All right, so this is what that nucleotide is going to look like. We have our phosphate here on the left, our deoxyarabinose in the middle, and then we have our guanine base connected to that carbon. All right, so in this one, we're going to take a look at how we sort of orient ourselves when it comes to the carbons that you have in the sugar part of your nucleotides. Remember, we saw this in the last question as well. This is going to be carbon number 1, and we refer to it as the 1' position. The prime is used to differentiate it from carbons in any other parts of the structure, right? So when you see the prime right after the number, that means that it belongs, that particular carbon is on the sugar. So this is our 1', 2', 3', 4', and this one here is the 5' carbon, right? That's off of the ring. It says to replace with a chlorine atom all the substituents except hydrogen on the 5' carbon. So there's my 5', I have two H's attached to it, and I have this phosphate group attached. I'm going to replace the phosphate because those aren't H's. I'm going to erase those. And I'm going to replace it with a Cl. All right, so the bond there now is to a Cl. See if I can draw that in right here and find a chlorine. All right, and there you go. All right, so you need to locate the correct carbon in your structure and replace, in this case, anything other than an H with a Cl. All right, so in this question, we're taking a look at how the bases pair up in DNA. And in these questions, you're given half of a base pair of DNA, and we need to draw the rest of it, and we need to connect them via the glue that sticks them together, which is the hydrogen bonding, right? So remember, in DNA, the DNA double helix is held together by base pairing, complementary base pairing, which is very important. It allows us to do lots of things, including predicting the sequence of bases. So in DNA, your cytosine pairs up with your guanine, and then your adenine pairs up with your thymine, right? That's the base pairing that happens. C goes with G, or G goes with C, A goes with T, or T goes with A. Now, the number of hydrogen bonds that connects them is different in each case because of the structures. G and C are connected via 3 hydrogen bonds. A and T are connected via 2 hydrogen bonds. Okay, so the first thing we need to decide is which base is shown. So you need to look up on your data sheet the structure of the structures of the bases to identify what they are. Okay, so in this case, we can see that that structure looks like the structure of cytosine. That's this one. If you take it and flip it, right, if you just take this and rotate it around, that's what we have here. Okay, so that's cytosine and it's on the right side. What does cytosine pair up with? Well, it pairs up with guanine, right, G. So we need to find the guanine structure, draw that over here on the left. There's guanine, draw it on the left side, and we're going to connect now them via hydrogen bonds, right? So what is that going to look like? Well, if we go back to and again, you can find a lot of this in your text as well. We want to look at GC base pairing because we just identified the cytosine, and there's an example of what you would want to be drawing, right? So you're provided with the structure of cytosine already. We identified that. You would need to draw in guanine in the right orientation and then show your hydrogen bond between, in this case, the O and the H here at the top, the hydrogen and the nitrogen there in the middle, and the hydrogen and the oxygen here at the bottom as well. In your template, you will see that there is an option, or there should be an option for adding hydrogen bonds. Okay, so make sure that you use that when you draw it, and it'll look something like this when you hydrogen bond, right? So you need to have another atom over here to hydrogen bond that too. So that's how you add H bonds in your structure. Again, you need to have your guanine drawn in and then connect the atoms to the atoms on this side via those H bonds. All right, so predicting the DNA-based sequence is something that's probably one of the simpler things to do in this section, because as long as you know what the rule is for base pairing, you can predict DNA-based sequences on that. So we know that C goes with G, or G goes with C, and then A pairs up with T, or T pairs up with A. So if you're given this sequence, we can go ahead and type it in the order that they will pair up with. Right, so you'll have an A, a G, A, and then T, and then an A, right? So this here is complementary to what we see that's provided in the same order of the same sequence. All right, so in this particular section, we're looking at nucleic acids generally, and you'll see DNA, you'll see RNA as well. And in RNA, there are actually a couple different types. There's 3 types of RNA. You have messenger RNA or mRNA, you have transfer RNA or tRNA, and then you have ribosomal RNA or rRNA, and they are found in different locations, and they do different things in the cell. So in this particular problem, you're given the description or a diagram, and you have to identify what type of RNA that thing is. So it says, consider the molecule X, which is this thing here. What is the name of X? And the answer should be this, one of these options for RNA, right? So what's going on in this particular diagram? Well, here you can identify your messenger RNA, right? That's your transcript. And then you're actually told this is the ribosome that you have. Right? So the ribosome has a large subunit, has a small subunit. And the ribosome is where protein synthesis happens. So what are proteins made up of? They're made up of amino acids. So in order to make a protein, you need amino acids being connected together. What brings the amino acids to the ribosome? Well, that's your transfer RNA, right? These transfer amino acids here at the top to the site of protein synthesis, where they can be connected via these peptide or these amide bonds, and you have your protein being made, right? So in this case, that would be your transfer or tRNA. So in some cases, you're provided with a diagram, other cases, you're provided with an actual description. All right, so in this question, you're provided with, or you need to figure out the relationship now between DNA and the mRNA that can be transcribed from it. So you're provided here with an mRNA sequence in a 5' to 3' direction, and we need to figure out the base sequence of the template strand, also called the antisense strand of DNA, from which it could have been transcribed. All right, so remember now, in transcription, right, that's the process whereby DNA, specifically the template strand, is converted into messenger RNA. Okay, so that means in order for the mRNA to be made, this is going to be complementary to the template DNA strand. So all we need to do is to use our complementary base pairing to figure out what's going on here, right? So in this, in the template, or the antisense strand is going to be in the opposite direction. So it's going to be 3' to 5'. Okay, all we're going to do is figure out now what C pairs up with, right? So as we saw before, C pairs up with G. We have 3 of those. So CCC will pair up with GGG. The G now is going to pair up with the C. U that you have in the messenger RNA pairs up with A. In DNA, you have another C that would be pairs up with a G. A in messenger RNA will pair up with a T in DNA. So remember, in DNA, one of the main differences between DNA and RNA is that in DNA you have thymine. T in RNA, you don't have thymine, you have uracil. Right, so A will pair up with a T in DNA, and then C will pair up with a G. And that's it. That's going to be the complementary base DNA base sequence on the template strand, complementary to this mRNA transcript. All right, so now you're given that mRNA transcript again, and what we need to do is to translate that message into an actual peptide. And specifically, we need to figure out what are the amino acids that are going to make up the peptide that's created. So we're going to read this in the 5' to 3' direction, and we're going to read it in groups of 3, right? So it's a triplet code that we're looking at here. So we have 5 prime, it goes A, U, G, and what I like to do is to write it out and then separate it in groups of 3 so we can actually look up the genetic code, which is a 3-letter code anyway. And we have GCC, we have GGA, that's a G, we have UGG, where are we? UGG, we have GAC, and then UGA. All right, so separate them out, and you can rewrite it on a sheet of paper and separate them in groups of 3. That would probably be the best way to approach this. And then we're just going to use a genetic code. So you don't need to memorize the genetic code. You can look it up, and you'll be provided with it on your exam. So let's scroll down here, see if we can find it. There's a genetic code. And we're just going to interpret it, right? So we're going to start at the front end, the 5' end, AUG, always. So there's A, that's the first base, U, the second, and then G here. That lines up to methionine, so MET. We want a 3-letter code. We're going to separate it with a dash. GCC codes for what? Let's see. So GC, C would code for alanine. That's ALA. GGA codes for what? G, that's the first base. The second base is a G, and then A here would also be, this looks like it's GLY, which is glycine. UGG codes for what? Let's see, there's first letter is U, second is G, and then the third letter G is TRP, which is tryptophan. GAC codes for, so there's G is the first letter, A is the second, C is ASP, which is aspartate. And then UGA, so UGA is a stop codon. And it looks like that. So we're going to type these in now. So MET, methionine, dash or hyphen ALA, GLY, TRP, ASP, and then stop. So let's see if there's any information about how we entered the stop codon. Doesn't look that way. So let's see. All right, so let's see what it says here. So we don't need to actually put the stop codon in, okay, because it's when the translational machinery encounters that, it'll automatically release that that peptide, right? So that's where we're gonna stop our list. So methionine, alanine, glycine, tryptophan, and aspartate. So that's a 1, 2, 3, 4, 5. That's a pentapeptide.